Okay, perfect. Good morning, everyone. Good afternoon and good evening. Welcome to the Think Tank table. We are here located in Princeton. We also have the Boston hub on screen, as well as the thinkers all around the US and some also ex-US as well. Thank you for joining. This is June the 30th. And so we thought what better way than to kind of have a review where the data set is in terms of all the different malignancies that we saw in the last 6 months timeframe. Of course, ASCO is a great milestone in terms of where the data sets are. The thinkers are on the table. All of us each individually, we own a pillar. So we're happy to discuss some of the changes and the paradigm shifts or lack thereof that we have seen in the last 6 months. Happy to discuss with you on your thoughts, questions, and comments. Every single panel, after the 3 different sets of discussion, Mahim, Mujia, and the team and I will pause and we'll discuss and take your questions. So please feel free to send in your questions, comments, or something that you don't agree with. With that, Mahim will walk us through some of the changes that we have seen in the last 6 months and some changes we are projecting that is yet to happen in 2022.
Thank you so much, Hafiz. So as Hafiz mentioned, each of us will go through tumor types, what are the key updates that happened in first half, and what are some of the trends and implications that we are expecting in that tumor type. Before I pass it on to Andrew to walk us through the lung tumor type, I just wanted to give a quick overview of key themes that we are observing across tumor types. So starting with checkpoint inhibitors, as everyone on this call I'm sure are aware that checkpoint inhibitors have transformed the treatment landscape in metastatic setting, but they are definitely expanding their presence in early stage disease as well. And you will see some of those examples in lung, where Opdivo got approved in neoadjuvant setting, and then we know there has been data from other checkpoint in adjuvant trials as well. And the same goes for breast, bladder, RCC, and melanoma as well. The second key theme is around antibody drug conjugates. Again, very familiar topic. These are gaining prominence across tumor types. Similar kind of trend, they are approved and have shown incredible activity in second line, third line, so late line metastatic setting. And now each of these ADCs have plans to move to early lines of therapy, particularly in first line, either as monotherapy to replace chemotherapy backbone or in combination with current standard of care as well. The third theme is around, unfortunately, underwhelming results from novel immuno-oncology mechanism of action. Particularly, I wanted to call out TIGIT and IL-2. Of course, it's not the end of story, and I'm sure Andrew will touch on it a little bit in more detail as well. But there were some underwhelming results, particularly in small cell lung cancer, and then we are expecting to see sort of long-term overall survival data in non-small as well. Similar, a little bit more negative for IL-2, where one of the assets which were in late line in phase 3 had to be discontinued because of underwhelming results. Last but not the least, again, one of our team members, Fiona, will discuss this as part of precision medicine. But as we are all aware, liquid biopsy has been explored in multiple settings across the disease spectrum. So from disease monitoring to disease diagnostics to even selecting patients' prognostic implications as well across tumor types, but particularly much more advanced in lung, CRC, and heme. On that note, I'll pass it on to Andrew, who will walk us through the key data and some of the trends in lung cancer.
Great. Thanks very much, Hafiz, and thanks very much, Mahim, for the introduction. As you see here, 2022 has been an impactful year in lung cancer. And standing here in late June, there are a few key events we've seen thus far and a few that we're still expecting later on. These broadly fall into 3 main buckets, which are consistent with the themes that Mahim just mentioned. The first is the spread of checkpoint inhibitors to earlier stages of lung cancer. The second is the emerging role of novel IO in lung cancer and the open questions that remain. And the third is new data for targeted therapies, reinforcing the need to test for actionable biomarkers. So let's first unpack IO moving into neoadjuvant and adjuvant. In the neoadjuvant setting, we saw the FDA approval of nivolumab and chemo in March 2022. This was based on EFS, and it was the first IO approval in neoadjuvant lung. A few weeks ago at ASCO, we saw data presented for the TIGIT class, as Mahim alluded to, importantly in small cell lung cancer, not NSCLC. So small cell is a notoriously tough disease to see an efficacy signal, And what we saw here with Skyscraper 02 on the bottom left-hand side here was tiragolumab and atezo plus chemo failed to meet the PFS endpoint. That said, the TIGIT story is not done. It's still early days. And this is a crowded space for novel IO with TIGIT inhibitors from Roche, Arcus, and Merck, just to name a few. The non-small cell study we have our eyes on is the phase 3 Skyscraper 01, where Roche failed to achieve statistical significance in PFS in the PD-L1 high expressors, but most of the alpha or statistical spend was in OS, overall survival. So questions remain about how the survival benefit will net out across PD-L1 expression levels. If we look to the top right-hand corner of this slide, we'll transition to the discussion to targeted therapies and the ever-evolving KRAS G12C class. Here we're expecting to see the CodeBreak 200 readout. That is randomized data of sotorasib, which is a G12C inhibitor currently approved for lung, versus docetaxel. And there are open questions about the benchmark, the efficacy benchmark of docetaxel post-IO. As a reminder, sotorasib already has accelerated approval, and this is expected to support full approval. Looking at the G12C class in general, adagrasib has a PDUFA date later in December of this year and has demonstrated combo data with pembro, as well as data in active CNS Mets. Sotorasib will be showing combo data with pembro and combo data with SHP2 at World Lung later this summer. If we transition to the bottom right-hand corner of this slide, we would like to spotlight Keynote 91, which is for adjuvant lung. Roche already has an approval with Empower 10, but the key play here is DFS benefit regardless of PD-L1 status. So with that, we'll move on in lung. And as Mahim mentioned, these are sort of the key themes which were teed up before. There's some skepticism regarding novel IO, but the story is still evolving. ADCs are slated to enter the lung cancer treatment paradigm, and the competitive environment is further increasing with KRAS G12C. So with that, I'd like to pass it on to Marissa to discuss some of the key themes in breast cancer.
Thank you so much, Andrew, for that overview of lung cancer and the excitement there. Breast cancer has been quite exciting itself in 2022. Starting off in Q1, we saw the FDA approval of olaparib, the first PARP inhibitor in the adjuvant setting for gBRCA mutated HER2 negative early breast cancer patients based on the remarkable results of the Olympia trial. Recently at ASCO in this quarter, we saw some very exciting results from the DESTINY-Breast04 trial, showing substantial improvement with HER2 in both PFS and OS over physician's choice chemo for this new category of HER2 low metastatic breast cancer. And right now, this is being defined as patients that have IHC HER2 1 positive or 2 positive with also IHS test negative. And right now we're expecting approval for HER2 in this setting by the end of the year. And also looking towards the second half of the year in Q3, we're expecting follow-up data from monarchE. So earlier this year, monarchE was approved in the adjuvant setting for high-risk HR positive patients with a high Ka67 score greater than 20%. However, with these follow-up data, there's the potential to broaden the current approved indication for abemaciclib in HR positive disease and further expand the adjuvant CDK4/6 to now the broader high-risk patient population in the United States. And we've already seen this happening in ex-US markets such as the EU, Brazil, Japan, where this drug is available for patients without the Ki67 restriction. And lastly, looking towards the end of the year, we're expecting data from Novartis's trial, the NATALY study, also investigating another CDK4/6 inhibitor, ribociclib plus endocrine therapy. And interestingly enough, this study is looking to, again, expand the CDK4/6 space in the adjuvant setting, now to include intermediate risk patients. So moving on, we wanted to dive a little bit more deeply into the DESTINY-Breast04 trial results because of how impactful and incredible these results were. Looking across the board, we see remarkable PFS and OS benefit over TPC in this new HER2-low subtype, with hazard ratios of approximately 0.5 to 0.6 across the board for both HR-positive and HR-negative patients. And looking to the left-hand side of the screen, we see some impressive Kaplan-Meier curves with great separations for all patients, regardless of HR status. And with such impressive results and high physician enthusiasm with these results, we're really seeing Enhertu gaining this potential to redefine treatment for a significant amount of metastatic breast cancer patients, approximately 55 to 60% of patients that are currently classified as HER2 negative, but now will be reclassified as this new HER2 low subcategory. So moving on to discuss some of the larger trends and implications that we're seeing for 2022, there's a lot of themes that come to light here for breast cancer. First, as we just spoke, is the emergence of HER2 low metastatic breast cancer. Really, the strong results from Destiny Breast 04 have resonated with these physicians, or many are eagerly trying to retest their patients to make sure that their patients can qualify for Enhertu. But now some important questions are coming to light, such as how do we optimize testing methods for HER2 low and eventually new HER2 ultra low? And what do we do now with treatment sequencing with the different ADCs that we have and different targeted therapies? Also, more largely, we see that ADCs are taking on a larger role in breast cancer, from Enhertu in the HR positive space to now the HR low space to sacituzumab and TNBC, and now HR positive space, and now sacituzumab expanding into earlier lines of therapy. And we also see new agents such as HER3 antibody drug conjugates also making a big splash in breast cancer. And finally, we're really looking forward to understanding the differences between CDK4/6 inhibitors in the metastatic and early HR positive breast cancer space. Right now, there's a lot of confusion among physicians as to why there have been so many different results between different CDK4/6 inhibitors in the early setting and also the metastatic setting. So physicians are looking towards the NATALEE trial that we spoke about to help elucidate some of these differences and help key down whether these CDK4/6 inhibitors are actually have various properties or not. So now I'm going to pass it over to the GYN team, to Chirant, to share with us some details on GYN from this year.
Hi, my name is Chirant, and I'll be going over some of the recent data developments in GYN. 2022 has been an interesting year for GYN with multiple pivotal readouts across ovarian, cervical, and endometrial cancer. Starting with cervical cancer, in March, the phase 3 CALA trial evaluating durvalumab with chemoradiotherapy in locally advanced cervical cancer failed to meet its primary endpoint of PFS compared to chemoradiotherapy alone. This is significant because the role of IO is being actively investigated in this setting, and CALA being the first trial to read out could provide some additional insights for ongoing and future trials. Moving to ovarian cancer, benefit with PARP maintenance in first line was reinforced by yet another positive phase 3 study. The results from the pivotal Athena mono study was presented at ASCO, and the PFS results support the use of rucaparib maintenance in first line, regardless of HRD status. As many of you would be aware that the discussions for our regulatory filing is still ongoing, but nonetheless, an approval is expected, which would make rucaparib the third PARP inhibitor to enter the space after niraparib and olaparib. Shifting gears to some of the upcoming events, the data from the part 1 of a Ruby trial evaluating dostarlimab with chemo in first-line endometrial cancer is expected to read out in the later half of the year. It is important to note that a Ruby 1 is the first IO plus chemo trial to read out in this setting. And finally, by end of the year, folate receptor alpha targeting ADC mirvetuximab is expected to receive accelerated approval in platinum-resistant ovarian cancer. The FDA has set November 28 as the PDUFA action date. This is particularly exciting because not only is Merck the first ADC to be approved in ovarian cancer, but it is also providing an additional option to patients in a difficult to treat a disease setting. So thinking about some of the trends and implications, ADCs like tisotumab vedotin in cervical and mirvetuximab in ovarian cancer have proven to be effective therapies in late-line setting. And based on some of the recent data, they could move to earlier lines of therapy with or without standard of care. Furthermore, in endometrial cancer, there is increasing use of biomarker-directed approaches, which is likely to transform the first-line treatment landscape, where ongoing pivotal trials are focusing on molecular subtypes, namely MMR, MSI for IO monotherapy, P53 wild type for selinexor, and HRD status for PARP therapy. This is pursued in order to assess benefit in these subpopulation as well as to support their approval. And lastly, autologous TIL therapy could enter the cervical space after their success in melanoma. This is particularly because of the encouraging activity that has been observed in the metastatic disease thus far. In any case, the approval is expected sometime in 2023 next year. Thank you.
Awesome. Great. Thanks. Thanks, Shiran. I'll like to invite Andrew and Marissa back to the panel to have a quick discussion. And I wanted to encourage all the attendees who have joined us. First of all, thank you for joining. And second, please use the Q&A feature if you have any questions or any comments that you would like us to discuss during some of these mini panels. So Andrew and Marissa, if you don't mind, if you can come back and turn the videos on, and then we can have a quick sort of discussion on some of the questions that we are seeing from the attendees.
Great. So if I may go first, Andrew talked about TIGIT a little bit. And so in terms of Skyscraper 2, is there still hope for TIGIT in lung?
So cautiously, yes. Just because one TIGIT candidate failed to meet PFS in one trial in small cell doesn't mean it's over for lung in general. You got to remember that non-small cell lung is a very different disease from small cell. And in non-small cell, we saw encouraging phase 2 data for atezo and tiragolumab in Cityscape at ESMO IO last year with OS not reached. So that said, when we look at non-small cell, there's also Skyscraper 01. And even though they failed to show PFS in PD-L1 high, most of the alpha was for OS, remember. So there is still hope for overall survival in non-small cell, but there's a lot really riding on that readout. And broadly, there are questions about the contribution effect of TIGIT relative to the existing IO backbone. So in short, time will tell for this class.
Great. I think there's a question on ADCs coming up.
Yes, so there is a question for Marissa. You mentioned a couple of ADCs. You mentioned how NR2 had HER2 low, sacituzumab has approval in TNBC and some data in HR positive. And they might have plans to move in early lines of setting. Do you see any hurdles or challenges as they move up in the line of therapy in first line or even early stage setting?
Yeah, so as you mentioned, sacituzumab, all these drugs are definitely moving up. And one of the issues right now that they're facing in the earlier lines of setting, especially in the adjuvant setting, is some toxicity challenges. We saw at ESMO Breast, sacituzumab is being explored in the post-neoadjuvant setting in the Saskia trial. Unfortunately, there are larger cases of myeloid suppression, heme toxicities, non-heme toxicities that were far more than physicians would like to see. So they're really hoping that as sacituzumab and other ADCs move up in lines of therapy, that they'll be able to show enough efficacy to really justify this higher toxicity profile that some of these ADCs are showing.
Great. No, thank you. Awesome. Thanks, Andrew. Thanks, Marissa.
And I have a question for you.
Oh, yes.
Given you're the in-house gyne expert at the table, right? So what are some of the IO entry point into ovarian?
Yes,
Is there hope?
Yeah, so ovarian cancer has traditionally been termed as one of those quote unquote cold tumors. Immuno-oncology, different PD-1, PD-L1 inhibitors have been evaluated in line setting for monotherapy, but the response rates haven't been as impressive as we have seen in other tumor types like lung or melanoma. The response rates have been in the range of 10 to 15%. But yes, the hope is still not lost. There are ongoing trials where the expectation is that the combination of PD-1, PD-L1 inhibitor with PARP inhibitor can increase their activity synergistically. There are some mechanistic rationale to believe that this combination can help immuno-oncology enter ovarian cancer. So we will see, and we are expecting some of those trials to read out either late this year or early this year. So we can expect some immuno-oncology love in ovarian as well.
Perfect. It's good for patients. So in the interest of time, we'll jump right back into prostate with Madeline.
All right, thank you so much, everyone. My name is Madeline, and I keep up with all the exciting prostate cancer developments at Axiom. And this year, we know there has been a lot of activity in the field. So we've seen the approval of new MOA, some key readouts for targeted therapy in non-targeted patients, and also the treatment intensification in earlier stages. So just sticking to the highlights, at ASCO GU in February, we were excited to see the first phase 3 data on PARP plus NHAs showing benefit in all comers with PROPEL, but only in HRM with magnitude. So this has sparked a debate on whether this targeted agent is really for everyone or only for those who present certain biomarkers. So it'll be really interesting to see how the story develops. And also in Q1, we saw 177 lutetium PSMA, also known as Pluvicto, which was approved based on amazing vision results for late-line patients with really limited treatment options. So this approval has been met with a lot of enthusiasm among both patients and physicians. And moving on to Q2, at ASCO, iPotential 150, which was looking at AKT inhibitors for PTEN loss, and therapy, which was a smaller phase 2 study comparing 177 lutetium PSMA versus cabazitaxel, we saw that neither study had statistically significant OS benefit, but in the case of radioligand therapy, since it had better patient-reported outcomes and RPFS benefit, we can still expect it to be sequenced prior to cabazitaxel. So transitioning to Q3, we are expecting another PARP and NHA readout at ESMO with TALAPRO-2, which really has the potential to impact perception of PARP-based therapy and its role in HRM only or in a broader biomarker agnostic population. And finally, in Q4, we are hopefully looking forward to the approval of triplet therapy in MCSPC based on pivotal ARISON's data, which showed 32% reduction in risk of death for patients who received this treatment intensification. So moving on to trends and implications, looking at key trends seen this year, PSMA PET scans are anticipated to become standard of care as it was included in NCCN guidelines this May, and there is high KOL enthusiasm for this next generation imaging. But before widespread uptake, there are some hurdles, including the fact that commercially approved agents have only recently been launched, as well as some logistical and reimbursement challenges. And with new imaging, there's a whole new set of questions. So we don't know how upstaging patients based on more sensitive imaging will actually impact treatment patterns and survival. So we'll be paying close attention to how physicians use this technology. And our next big bet is treatment intensification in earlier settings and novel MOA in later settings, which means we predict NHA-based regimens moving into earlier lines of therapy, either in combinations with chemo and MCSPC, or into the high-risk non-metastatic setting, potentially with or without ADT. And as for novel MOAs, we think more PSMA radioligand therapies and IO combinations are going to enter MCRPC. And finally, we are seeing a trend of targeted therapies being open to a broader patient population when used in combination with an NHA. So while we know that testing is important for treatment selection and sequencing, especially since PARPs are only approved in HRM or BRCA patients, it will be interesting to see if there are synergistic effects that benefit all patients, or what biomarkers besides BRCA are strong responders to PARP. And with that, I'll pass it off to Caleb for bladder updates.
Thank you, Madeline. Compared to the rest of the pillars, bladder cancer has been relatively quiet this year, with a few key events that have helped shape its future. Earlier this year in April, enfortumab vedotin was approved in the European Union based on phase 3 trial data from EV301 that demonstrated an overall survival benefit compared to chemotherapy for third-line metastatic patients that had previously failed platinum-based chemo and a PD-1 or PD-L1 inhibitor. This approval came after much concern and discussion surrounding the skin toxicities associated with EV, but ultimately the benefit was decided to outweigh the risks, resulting in EU approval, bringing EV to the global stage within the metastatic setting. The next month, we witnessed a top-line data readout from Checkmate 901, which investigates Nivo plus IPI and Nivo plus chemotherapy combinations in the first-line metastatic setting. This readout indicated that Checkmate 901 failed to meet their OS primary endpoint in the PD-L1 positive patient population for their Nivo plus IPI arm. And expectations for this IO-IO combination were once hopeful, given that there was benefit previously demonstrated in Danube's PD-L1 positive patient subgroup. However, given this most recent failure, expectations for the success of the remainder of Checkmate 901 are pretty low, marking chemotherapy as the continued standard of care in the first-line setting. Also in May, ImmunityBio filed a BLA for their IL-15 superagonist N803 plus BCG for BCG unresponsive non-muscle invasive bladder cancer patients. This filing was based on the overall positive efficacy and safety results of the QUILT 3032 CIS cohort A and has a high potential for approval. In regard to what we're hoping for later this year, in September at ESMO, we do hope to see an OS data readout for IMvigor130, investigating atezolizumab plus chemo or atezolizumab monotherapy versus chemo in the first-line metastatic setting. These data are long awaited following the initial announcement in Q3 of 2019, stating that this trial did meet its co-primary endpoint PFS. Based on analogs and timing since the initial PFS readout, expectations for positive OS in this IO plus chemo combination are really not that high, is what we're noticing. This leads me to my key trends and implications in bladder cancer for 2022. We can go, yep. First, with IO plus IO and IO plus chemo combinations having faced many challenges in the first-line setting, thus far we've seen multiple trials fall short in providing benefit over chemotherapy in the first-line setting, and expectations for these combinations appear to be lower than ever for similar trials like IMvigor130, like I mentioned before, NILE, and Checkmate 901's remaining endpoints. Secondly, there appears to be a growing role of antibody-drug conjugates across bladder cancer. ADCs are being explored to their fullest extent, with a shift in focus in bringing ADCs from where they reside now in late-stage bladder cancer to earlier stages as mono and combination therapies. We've also noticed within the ADC realm that there's an emerging development for HER2 targeting ADCs in the first-line setting. Lastly, there seems to be more development and success for intravesical therapies in the BCG unresponsive NMIBC setting, with intravesical agents like N803 or CG Oncology CG0070 alone or in combination with immunotherapy that are demonstrating positive efficacy and safety and potentially shifting the NMIBC paradigm to a future of intravesical combinations. With that, I conclude the bladder review, and I'll pass it off to Oren for a review of RCC.
Thanks, Caleb. And hi, everyone. I'm Oren, and we'll be discussing key events and trends in the RCC space. And while the first half of 2022 has not necessarily highlighted any game-changing data readouts or approvals in RCC, we are in fact expecting several registrational trial readouts in the second half of 2022. Looking at the past few months, however, RCC has seen several promising therapeutics with novel mechanisms of action fail to demonstrate a meaningful benefit versus the current standard of care therapies. In April, the much anticipated Pivot-09 trial investigating nivolumab plus Bempeg, a novel interleukin-2 immunotherapy, failed to meet its pre-specified primary and secondary endpoints in frontline RCC. Looking ahead to the second half of 2022, The first registrational triplet trial, Cosmic 313, investigating Nivo plus Ipi plus Cabo in frontline RCC, is expected to read out top-line data shortly in July, followed by full data presentation at ESMO this year. This triplet regimen has the potential to greatly disrupt the current treatment paradigm by raising the efficacy bar over the current standard of care immunotherapy doublet combinations. And finally, looking to the bottom of the slide, CONTACT-03 is a phase 3 trial investigating atezo plus cabozantinib in second-line plus patients, and will answer key treatment sequencing questions related to the optimal later line therapy selection following progression on immunotherapy in the front line, an area currently lacking randomized data to inform clinical decision-making. Moving on to our key trends across kidney cancer. In the past couple of years, we have seen IO growing in dominance throughout lines of therapy in RCC. It has now almost entirely displaced TKI monotherapy in the front line. The frontline RCC landscape is currently hyper-competitive with 4 IO-TKI combinations and 1 IO-IO combination as standard of care regimens. Immunotherapy has also recently entered the adjuvant setting, as after a multitude of failed adjuvant TKI trials, Pembro demonstrated a highly significant efficacy benefit in high-risk localized RCC patients. In the next couple of years, we also expect to see some data on IO retreatment in the second-line plus setting. Circling back to the front line, we see a trend of looking to triplets to reach the next level of efficacy. Cosmic 313, testing Nivo, Ipi, and Cabo, will be the first of several triplet regimens to release data and will shed light on whether triplet combinations are the future, particularly whether there is sufficient efficacy benefit to justify the expected increased toxicity. Another strategy to improve efficacy is through investigating biomarkers and molecular drivers of disease to better tailor treatment decisions to each patient. While PD-L1 testing has historically not been a useful predictive biomarker for RCC, we are seeing a continuing search for useful biomarkers to assist in treatment selection, including in the adjuvant setting. And finally, as somatic VHL gene mutations are key drivers of disease for many clear cell RCC patients, HIF-2 alpha inhibitors are the source of much anticipation and intrigue. And now passing it back to our moderators, Mahim and Hafiz, for some follow-up questions.
So thank you so much, Oren. And again, I'd like to request Caleb and Madeline to come back so that we can answer some of the questions that we are seeing in the Q&A session. Wajia, would you like to start the discussion? There is a question.
Yeah, there is a question on bladder, which is like, in light of the recent failures in first line especially, what is the expectation surrounding new data, and how do you think the standard of care will evolve in the coming few months?
Yeah, so with the recent failures in the first line uterine bladder cancer from the Checkmate 901 trial not meeting their OS primary endpoint for PD-L1 positive patients, the expectation for IO plus IO was highest in this particular subgroup. So now the remaining trials that are expected to read out in Vigor 130 and Nile, like I mentioned, and the rest of Checkmate 901, the expectation for success from these trials is pretty low. But where we can see that physicians are shifting their attention to is, instead of IO, rather IO plus ADC being enfortumab vedotin and pembrolizumab. It's where the most excitement is, and where we've been noticing that physicians think the most success will be in the first line metastatic setting.
Thank you.
So I know we only have a couple of minutes. Oren, what does the future hold regards to systemic therapy for localized RCC? You mentioned some of the IO data and approval in adjuvant setting, but curious to hear what do you think the future trends or implications could be for localized setting?
Yeah, absolutely. The future appears, quite bright for adjuvant therapy in RCC. There are several more ongoing pivotal trials aiming to move immunotherapy into the localized disease setting. So we have atezo and nivo monotherapy data sets expected to read out shortly in the near term, while IO combinations of nivo plus ipi as well as pembro plus belzutifan aim to continue to raise the efficacy bar over monotherapy. And then lastly, another really important point here, in light of the lack of biomarkers in RCC, and given the wide range of recurrence rates following surgery for these patients, the optimal patient selection for adjuvant therapy is a key area to continue improving on and trying to incorporate into clinical trials, and is really top of mind for physicians, given that the risk of overtreatment is really a critical consideration. So we'll continue to see how this field continues to progress, but overall should be very exciting to keep up with developments.
Awesome. Last question, there is one question on the Q&A, Madeline, for prostate. You mentioned how the radiopharmaceutical have shown impressive results in prostate cancer. Are there any hurdles, or what are your sort of insights on the feasibility of radiopharmaceuticals in real world?
Yeah, so this is definitely a really relevant question. As we've seen, there were supply shortages with Pluvicto that Novartis announced early in May, and this has been resolved somewhat, but due to the complexity of manufacturing radiopharmaceuticals and also the complexity with patient care. So it really takes a multidisciplinary team. So radiation oncologists, nuclear specialists, and medical oncologists all really working together, not to mention challenges with cost and reimbursement. So for 6 cycles of Pluvicto, it costs more than $250,000, which might not be accessible for everyone. So although there is a lot of excitement and enthusiasm surrounding this new mechanism of action and amazing data that we've seen in the VISION trial, so 0.4 RPFS hazard ratio, 0.6 OS hazard ratio, there are definitely some implementation hurdles that we will see how to address in these coming years.
Awesome. Thank you so much, Madeline. Thanks, Oren, and thanks, Caleb. Now I would like to invite Joel to walk us through the melanoma section of the presentation.
Thank you, Mahim. So turning it over to melanoma, in terms of events, earlier this year, the FDA approved a new first-in-class PD-1 LAG-3 co-formulation for the treatment of unresectable or metastatic melanoma. And this co-formulation is a fixed-dose combination of nivolumab and relatlimab, or Rela-Nivo, which was approved based on the phase 2/3 Relativity-047 study that compared Rela-Nivo to Nivo monotherapy in patients with advanced melanoma. In the Relativity-047 trial, the combination more than doubled the median PFS when compared to Nivo monotherapy, and importantly, this benefit was seen without added toxicity, as the safety profile of the combination was comparable to that of Nivo monotherapy, which I should note is much better than the safety profile of Ipi-Nivo, another IO-IO combination approved in this setting. As a result, the approval of Rela-Nivo is expected to potentially be used to replace IO monotherapy, at least in certain patients. It's also worth noting that the approval of Rela-Nivo is particularly significant because it introduces an entirely new combination of 2 immunotherapies that may act together to help improve anti-tumor response by targeting 2 different immune checkpoints. And we expect that the combination will also be evaluated in the earlier setting as well. More recently, we saw DMFS results from KEYNOTE-716, which evaluated pembrolizumab as adjuvant therapy in patients with resected stage 2B and 2C melanoma. And this regimen was approved by the FDA last year on the basis of the RFS benefit that was shown in the trial. At ASCO this year, we saw data on distant metastasis-free survival, a key secondary endpoint of the study, showing a significant improvement versus placebo in these patients. And as stage 2B and 2C patients previously went untreated, despite having a considerably high risk of recurrence, including distant metastases as first recurrence, these results lend further support to the use of pembrolizumab as the standard of care in patients with resected stage 2B or 2C melanoma. As a result, BMS is also evaluating their PD-1 nivolumab in the same patient population. The Checkmate 76 trial will likely read out at ESMO this year, and depending on the data, the results could expand the approved adjuvant indication for nivolumab to include stage 2B, 2C melanoma, and would continue to provide strong support for the need to treat these high-risk patients who have historically remained untreated after resection. In terms of trends, as exemplified by the approval of Keynote-716 and the initiation of other trials, on the next slide, the initiation of other trials like Checkmate 76K and Relativity 098, if my slide can just be advanced. There has been an increased interest in identifying those stage 2-3 patients who are considered high risk and would benefit from the treatment with the checkpoint inhibitor, either as monotherapy or in combination regimens. We expect that these trials will also lead to approval of novel regimens for patients who are still at high risk of recurrence. So novel combinations are also being explored in the advanced setting. Of course, melanoma has provided the foundation for IO-based treatment in oncology, and there are a number of novel IO combination regimens that are continuing to advance in the clinic. Beyond the PD-L1 LAG-3 combination, for example, other IO targets like TIGIT are also being evaluated in combination with PD-L1, of course, with the hope of seeing better success than it has seen in other tumor types like small cell lung cancer, as my colleague Andrew Hong noted earlier. Finally, approaches beyond anti-PD-1 are also being considered, and that's because despite being considered an immunologically hot tumor, many patients don't respond to PD-1 or are considered PD-1 refractory. And so novel approaches such as TIL cell therapies have already shown promising preliminary response rates in these patients, and we expect that some of these novel modalities will move beyond clinical development in the coming years. With that, I'd like to thank everyone for their attention, and I'll turn it over to my colleague Rohan Jidvani, who will discuss exciting developments in GI cancers.
Thanks so much, Joel. A major theme for gastrointestinal tumors so far in 2022 has been the continuation from last year of immunotherapy success as frontline therapy. The TOPAZ-1 trial, which added the durvalumab to the standard of care combo with chemotherapy of gemcitabine and cisplatin in frontline biliary tract cancer, produced significant improvement in overall survival, the first such for an IO drug in this setting, generating a 20% reduction in risk of death compared with gemcitabine alone. Notably, no additional toxicity from the checkpoint inhibitor was observed. Biliary tract cancers include cholangiocarcinomas and gallbladder cancer, which are rare, heterogeneous, and have poor prognosis. As a result, the FDA has granted the Durva combo priority review, so we can expect a launch in 2022. Among the positive results across gastric and gastroesophageal cancers over the past 18 months, the Checkmate 648 trial of nivolumab in combination with either a fluoropyrimidine and platinum-containing chemotherapy or with ipilimumab was recently granted FDA approval for frontline unresectable, advanced, or metastatic esophageal squamous cell carcinoma, agnostic of PD-L1 expression. The benefit in overall survival versus chemo alone produced a hazard ratio of 0.74, improved to 0.54 in patients with PD-L1 positive tumors. Benefit with the ipi-nivo combination was also observed, yielding a 36% reduction in risk of death among PD-L1 positive tumors. A fascinating preclinical presentation at AACR and ASCO highlighted a dual MEK inhibitor called IM1104, which has a selective pan-KRAS and NRAS activity. Clearly, this has application across a wide range of tumors, but data was able to show significant tumor growth inhibition in pancreatic models occurred, outcompeting the KRAS G12C inhibitors, sotorasib and adagrasib. And so the drug's ability to target the G12V mutation will be an important step forward for pancreatic cancer, where the G12C mutant accounts for only 2% of tumors, whereas G12V is significantly more prevalent. Given these encouraging results for a tumor with high unmet need, the agent is planned to move into a phase 1/2b trial that will include both pancreatic cancer and melanoma in Q4 following IND submission in Q3. Finally, positive results from the registrational Himalaya trial previously demonstrated significant improvement in OS versus standard of care sorafenib in frontline advanced HCC patients through the use of a single priming dose of tremelimumab followed by regular schedule of durvalumab. This novel IO-IO regimen received priority review in April and is expected to gain approval towards the end of the year. Turning to some data, one of the most talked about studies in recent weeks has been a small 18-patient trial evaluating the PD-L1 inhibitor dostarlimab in patients with locally advanced rectal cancer. Typically, patients with resectable disease can receive combinations of chemo, radiation, and surgery. These treatments, of course, particularly radiation and surgery, carry significant life-altering side effects, including bowel and bladder dysfunction, sexual dysfunction, and infertility, and permanent stroma in up to 30% of patients. And so the observation in the first 14 evaluable patients with a median follow-up of 6.8 months, that 6 months of dostarlimab resulted in complete remission in all patients, has the oncology community buzzing. It's critical to note that all patients in this study had the genotypic signature of mismatch repair deficiency, which is associated with positive outcomes for immunotherapy in other cancers and accounts for 5 to 10% of rectal cancer. The 100% confirmed CR rate has been hailed as unprecedented and points to the importance of genetic testing in early stages of rectal cancer. Given that 30% of patients in this setting progress on chemotherapy, points to a major clinical advancement for a chemo-free IO monotherapy regimen that is highly efficacious. The study will continue to follow the first 14 patients to evaluate durability of response, as well as report on the last 4 patients in the study who have also experienced responses and who continue on therapy. Perhaps the largest trend in GI cancer currently is the success of immunotherapy in frontline therapy, whether it is replacing standard of care, either by addition of a checkpoint inhibitor or through the use of checkpoint inhibitors alone, providing enhanced efficacy and or chemo-free treatment. Referencing the discussion around TOPAZ-1, genomic sequencing is increasing for cholangiocarcinoma given the increasing number of targeted agents showing success, including for FGFR, IDH1, BRAF, and MEK, as well as immunotherapy, even though benefit is seen in all comers. Ongoing trials in frontline metastatic colorectal cancer have attempted to fine-tune targeted therapy combinations. The triplet chemo FOLFOXIRI in combination with EGFR inhibition or bevacizumab has yielded improved overall survival to doublet chemo with either target agent. Finally, the next frontier in immuno-oncology is cell therapy in solid tumors. Advancements primarily in tumor targeting have enabled compelling results for CAR-T, including CD70 targeting for CRC, now in combination with Pembro, although that trial is currently on hold, as well as Claudin 18.2 targeting of gastric cancer patients who have failed multiple lines of therapy. Robust efficacy signals in late line has been observed in pancreatic cancer from an armored NK therapy that targets PD-L1 expressing PDAC tumors. And we expect to see an expansion overall of autologous and allogeneic cell therapy in other GI cancers over the coming years. So thank you, and I'd like to pass it over to my colleague, Gia, for some insights into heme.
Thank you, Rohan. Hi, everyone. I will be walking through some of the advances in the heme space, and we have a collection of different events across malignancies. So starting off with cell therapy in myeloma, this year we saw the approval of cilta-cel for relapsed refractory multiple myeloma. As a reminder, this is the second autologous cell therapy to be approved for these patients. And also at ASCO and EHA, we saw longer follow-up data that reiterated the benefit of this modality, right? Next, we saw the approval of ivosidenib for IDH mutated newly diagnosed older AML patients, and this is essentially a first-line approval from the use that was already there in certain other segments. And this was approved in combination with azacitidine. While these approvals are exciting, we also interestingly saw some withdrawal, notably of the PI3K class of inhibitors across different malignancies, and some of it was voluntary and some of it was prompted by regulatory authorities. Interestingly, we have seen this class of drugs being used extensively. However, it was determined that the data from overall survival analysis was not in favor of this class owing to the safety profile. And considering the fact that there are other alternates available to these patients, it was a logical next step to not use this class as of now. So we saw a few companies actually removing their agents from the market. We also saw the very exciting approval of mosunetuzumab, which is a first CD20 bispecific in the third-line space. And this is essentially the first approval for this class, and we have a number of different agents which are in different trials, and over the next couple of months, we should be seeing more activity in this space. Next, we move on to some updates from the myelofibrosis side of things, and we saw two events of interest over here. One was the data from momelotinib and secondly was the approval of pacritinib. And this essentially shows a slight shift in the way first and second line patients are treated from the standard of care of Jakafi, which exists as of now. Moving on to what do we expect coming forward, the biggest, most anticipated event or a series of events in heme is the approval of BCMA-based bispecifics, which should happen in the second half or even the first quarter of 2023. Notably, we have teclistamab and elranatamab, which are in different stages of regulatory review. And this will essentially be a new treatment choice for these patients. Lastly, we also expect the approval of zanubrutinib in CLL based on the data from the Alpine trial, and the NCCN recommendation for this use is already in place. Can we move on to the trends? Yeah. Moving on to the trends, I think the most notable trend that we see in the heme malignancies is bispecifics. It's a year of bispecifics. We've been seeing some amazing data come out from different trials over the last couple of years. And this year we will see approvals and clinical use in both myeloma and lymphoma. What this brings to the table is an increased efficacy benchmark, a good manageable safety profile, and most importantly, ease of administration. And this sets the standard for several patient segments. The second trend that I would like to highlight is cell therapy, especially the autologous cell therapies that have already been approved. We've been seeing increasing use in the later lines of therapy, but what we could expect in the next couple of months is more entrenchment of these approved therapies. Also, certain line movements that we are looking at, especially in the lymphoma side of things, wherein we are looking at certain approvals in place. Also, on the other patient segments, we will continue to see some data reiterating the role of cell therapy in either a second line space or in the first line space. Right. The last trend that I would like to highlight, and which kind of goes a little across different malignancies, is that shake-up of the first line options, and notably in the myelofibrosis, CLL, DLBCL space. We've had certain standard regimens or drugs that have been used over the years, and they've remained unchanged. But this year and the coming couple of months, we should see some shake-up in this space. As I mentioned earlier, the JAK inhibitors in myelofibrosis could see some new options come in. Similarly, chemo was primarily used for CLL first line, or rituximab-based therapies for DLBCL. And we should see agents like venetoclax, BTK inhibitors, polatuzumab, and the newer JAK inhibitors challenging this existing standard of care to bring about a better efficacy and safety benchmark for these patients. Thank you. And I will now hand it over to Fiona, who will walk us through some of the developments in precision medicine.
Thank you, Ajiha. This is Fiona. So now we're going to look at things from a more pan-tumor approach. I'll be wrapping up today's presentations with an overview of precision medicine key trends. So there has been a push for ctDNA use in the adjuvant setting, but currently we're still lacking in good and strong clinical evidence. In Q2, fortunately, we saw the dynamic trial presented at ASCO by Dr. Jane T. that supported the use of ctDNA as a tool to stratify patients in the adjuvant setting for colon cancer. At EHA this year, we also saw a very exciting trend. While cfDNA has been a relatively more well-established biomarkers in solid tumors, we haven't seen that as much in heme diseases. So at EHA this year, we saw a number of early prospective studies showing cfDNA's application in a clinical paradigm, either as a prognostic biomarker in predicting patient outcomes and or as an MRD biomarker to monitor patients' disease status. Another key theme of precision medicine as it relates to biomarker testing. In Q3 and Q4, we are expecting to see robust real-world studies that showcase testing rates presented at big congresses such as ESMO and ASH. Last year, a study at MyLung showed that only 50% of patients with lung cancer or CLL are tested for NCCN biomarkers and subsequently receive their recommended therapy. So building upon that finding, we're expecting to see more real-world data sets shedding light on the issue of testing rates. So a study we'd like to highlight is the dynamic trial. For context, surgery alone currently cures around 80% of stage 2 colon cancer patients. There is a need for precise recurrence risk prediction to limit the number of adjuvant treatment given to patients. The dynamic trial is investigating whether a ctDNA guided approach versus standard approach could actually reduce the use of adjuvant treatment without compromising recurrence risk. So the trial recruited 455 patients, and patients were randomized 2-to-1 into a ctDNA guided management or a standard management arm. For patients who have ctDNA positive, they received adjuvant chemo, and for patients who were ctDNA negative, they were placed in the observation cohort. This graph shows that the 2-year RFS rates are comparable in the two arms, in the ctDNA management arm and the standard management arm. But the proportion of patients receiving chemo actually decreased by half. So overall, this study supports the clinical utility of ctDNA to be adopted as a tool in guiding adjuvant therapy. Lastly, we would like to highlight 3 future trends in precision medicine. As mentioned in the dynamic trial, where we anticipate an increase in liquid biopsy use, particularly in the monitoring setting for patients receiving adjuvant therapy in early stage disease. We've seen the generation of large data sets across multiple solid tumor types aimed to support the use of these assays. Ultimately, this is expected to improve patient outcomes through earlier intervention based on molecular relapse. Following the launch of Gallery last year, the first commercially available pan-tumor early detection test, we expect to see more companies developing different early detection tests that explore different analytes, such as DNA fragmentomics and proteins, and may take a more tumor-specific approach to increase the sensitivity and specificity of these assays. Looking forward, if early detection tests are widely adopted, we're hoping to see a shift in cancer stage diagnosis to earlier stages, where patients have historically shown to have improved outcomes. Last but not least, in the heme space, we are also seeing more early studies supporting cfDNA's potential as a surrogate for tumor burden. cfDNA could be incorporated in the treatment paradigm as a prognostic biomarker or a biomarker for MRD monitoring along the patient treatment journey. So that wraps up our presentation section today for the mid-year review, and I'll hand it back to the moderators, Hafiz and Mahim, to wrap up today's session.
Great job. Thank you, Fiona. Let's bring the panel back in together. So Joel, in the interest of time, rapid fire here, quick question. So with the advances of LAG-3 in melanoma, what are the thoughts around IO monotherapy in advanced setting?
So yeah, the question is about patients who will receive IO monotherapy in the advanced setting. I think that's something that still has to be seen. There certainly does appear to be a patient population who should be treated with monotherapy, since they may not be able to tolerate the combination regimens. So I think at least those patients who are more frail or maybe older patients might do better with an IO monotherapy. I mean, these patients do very well even with monotherapy often.
Okay, great. That's great. Rohan, just a quick question. I know you're personally also very passionate about pancreatic cancer. There has been some recent failures in pan space. Are there any new MOAs or approaches that might change the unmet need in pan cancer?
Great question, Mahim. And insofar as new MOAs, it's somewhat of a tested MOA, but that has been gaining some attention, and that is CD40 agonism. And so in the phase 2 PRINCE trial, which was out of the Parker Institute, combinations of the CD40 agonist sodigalimab, nivolumab, and gem/nab-paclitaxel were evaluated in first-line metastatic PDAC and showing activity. So the nivo chemo arm showed a 1-year OS rate of 58%, which met the primary endpoint for statistically significant improvement over historical control. The interesting thing here is that very advanced multi-omic translational work is ongoing to further tease out predictive biomarkers for different permutations of this regimen. And so to date, the investigators showed that response to nivo chemo is associated with relatively more T cell based activation. And so CD39, PD1 positive T cells, T follicular cell helper cells, and TNF alpha signaling with low immunosuppressive biomarkers. And the Sotiga chemo arm, although it didn't meet statistical significance for superiority, it did show certain biomarkers that are associated with response. Notably, they had high levels of PD1, TBET positive CD4 T cells, along with Th1 and Th2 signaling, consistent with activation of B cells, dendritic cells, and other helper cells associated with the mechanism of CD40 agonism in the team. So overall, this trial was an excellent demonstration of the potential for biomarker-based predictive therapy, which particularly in a pancreatic cancer setting is something that may deliver success where the tumor is highly heterogeneous. I should also note that another CD40 agonist, SEA CD40, has showed promising early responses in an ongoing phase 1.
Really exciting that there is so much development in pancreatic cancer. So one last question to Fiona, who is our in-house precision medicine expert. So we saw that liquid biopsy can have application in different settings. Where do you think it has the most utility currently, or maybe let's say 5 years in future?
Sure, I think liquid biopsy applications are really exciting in the metastatic setting. So it could be a minimally invasive tool to monitor patients at a shorter time interval in a minimally invasive way, as well taking that a step further to stratify patients based on ctDNA level to receive treatments. So both our Axiom's precision medicine team and KOLs, I think we're all really excited about liquid biopsy's applications in the metastatic setting.
Awesome. Thank you so much, Fiona. Thank you, Joel, Rohan, and Wajiha for such an amazing overview of each tumor space. And now I'll pass it on to Wajiha to close this session with some of the highlights of what's coming next.
Thank you, Mahim, Hafiz, and the rest of the Axiom team for this very engaging session. Just want to highlight what's next from the Axiom space. We have a podcast called Axiom Catalyst that drops in every 2 weeks. And looking at the lineup of future speakers, we're looking at Professor Fabrice Berlesi, Dr. John Tsai, Dr. Patricia Loroso, and we look forward to you guys tuning into these podcasts on Spotify or on Apple Podcasts. Secondly, we'll also be at ESMO 2022 in Paris in September. Do come by our booth number 251, come say hi to us, and we will be there on site for this amazing oncology conference.
Great. And then if you're local in New Jersey or in the tri-state area, come join us on July 23rd at 8:30. It's a 5K. We are trying to raise funds for Cancer Patient Cycle Support Cycle 7, and we need your help. And if you're not able to join us for the 5K live, you can donate online. You can scan the barcode there. And thank you for joining this morning. We'll see you post-ESMO with another round of roundtable from the think tank. Thank you.